IDPs represent about a third of the proteins in the human proteome, and we know very little about them. We call them the Dark Proteome. And IDPs are involved in a huge number of functions in the cell. They control cellular regulation, signaling. They're involved in cellular organization of non-membrane bound compartments in the cell. They're closely associated with disease. So they're linked to cancer, diabetes, cardiovascular disease, infectious disease, neurodegenerative diseases such as Alzheimer's and Parkinson's disease. So they're incredibly important in human biology and obviously in medicine. And IDPs offer us new possibilities to develop novel drugs, novel therapeutic targets, and so they're extremely important to study and understand how they function. So they do pose many challenges, as I said. There's, as far as NMR, which is the central tool for studying IDPs, you can't use traditional structural biology methods such as crystallography because they don't have unique structures. They adopt ensembles of structures, certainly when they're in the free state, not bound to targets, but they frequently also adopt dynamic complexes when they interact with the target. And so the method of choice for studying them is NMR. Almost all the time we're coming across new and totally unexpected results. This concept of disordered proteins forming phase transitions and non-membrane compartments in the cell, for example, was totally unexpected. So there's a huge amount of work that needs to be done. Our particular work is beginning to provide new insights into mechanisms by which proteins bind to their targets, recognition processes, signaling pathways and signaling mechanisms. A lot of the complexity in signaling in a cell comes from disordered proteins and the post-translational modifications that they undergo. And so it's providing the technologies and beginning to scratch the surface of how these things are functioning. And ultimately, as we learn more about how disordered proteins function, what their role is in signaling and in disease, then it opens new possibilities for design of drugs and therapeutics. And part of our work that impinges directly on that is dealing with viral proteins. So many viruses express proteins when they infect a cell. They express proteins that are disordered and that hijack the disordered networks, protein networks in the cell. And so many of the DNA tumor viruses, for example, produce disordered proteins that will block the action of p53, the tumor suppressor. They hijack the cell cycle, and they do it by using their own disordered regions to mimic cellular disordered proteins. And we should be able to do the same thing with drugs. It's going to take an enormous effort to understand the role of these proteins, the dark proteome, its impact upon signaling in cells, organization of cells, its impact upon medicine. The sort of work that we're doing is providing some of the foundations of understanding of how IDPs function, how they function in signaling, how they interact with their binding partners, getting mechanistic insights. And understanding of these processes is going to be very important in the future for development of new drugs and therapeutics to target diseases associated with IDPs. As an example, one of the problems we're studying in the lab is how virus proteins, disordered virus proteins, function to hijack the cellular machinery. We're particularly interested in some of the DNA tumor viruses, human papillomavirus, for example, which causes cervical cancer, and trying to understand how disordered proteins produced by HPV and other DNA tumor viruses, once they infect a cell, how those disordered proteins interact with cellular partners and in competition with cellular proteins to hijack the cellular machinery. And the viruses have been very effective drug designers. Over billions of years, they have evolved to target the cellular machinery in ways that we can't. And I think this is a real lesson for us there in terms of understanding how viral IDPs function in the cell to guide us to design our own molecules to disrupt cellular pathways and treat disease.